This week's video is going to be about composition of blood. Blood is comprised of two main components, plasma and then formed elements. Plasma accounts for about 55% and is primarily composed of 92% water. Plasma is considered a type of colloid. The major things in this colloid are albumin, which is the most abundant plasma protein, maybe about 60% of all blood proteins. Globulin is the next most abundant plasma protein and is involved in transporting substances within the blood. And fibrinogen, which is an important clotting factor. The formed element component of the blood consists of red blood cells, white blood cells, and cell fragments known as platelets. Red blood cells are the largest contributor to the formed element portion, making up about 45% of the total blood volume. The percentage of total blood volume is the red blood cells that is coagulated as the hematocrit. White blood cells and platelets make up a much smaller percentage, are found in a thin layer called the buffy coat or the buffy layer between the red blood cells and the plasma. When we break down red blood cells lack a nucleus and other organelles, which gives them a limited lifespan, approximately 120 days once they're in circulation. To meet the body's demands, making red blood cells is something that's constantly occurring. Specific mechanisms are necessary to handle the disposal of the vast number of red blood cells that die each second. Old and damaged red blood cells are initially processed by white blood cells, which are found in the bone marrow, liver, and spleen. They're then broken down, and each part of it is divided into other things. One of the main things that it gets divided in is to biliverdin, which is converted into bilirubin and released into the bloodstream. Bilirubin binds to albumin proteins for transport to the liver. This form is known as free bilirubin. Liver cells take up free bilirubin and conjugate it by binding a molecule called glucuronic acid to it. This creates conjugated bilirubin, which is water soluble and becomes a component of bile. Bile is secreted from the liver into the digestive tract. In the digestive tract, glucuronic acid is removed by bacteria and bilirubin is converted to urobilinogen. Some urobilinogen can re-enter the blood circulation and cycle back to the liver. And some of it is filtered to the kidneys where it becomes urobilin, which is yellow and contributes to the color of your urine. Urobilinogen that remains urobilinogen that remains in the intestine is converted by bacteria to stereobilin, which is a dark brown pigment that contributes to the color of feces. Homeostasis is the process by which bleeding is stopped and which is vital for survival. It can be divided into three main steps. We're going to have a vascular spasm, development of the platelet plug, and the blood clot formation. Your vascular spasm is not detailed super extensively in this class, but it's just the constriction of your damaged blood vessels, which is going to reduce that blood flow. When you talk about platelet plug formation, platelets are denucleated cells derived from other things. They play an important role in blood clotting. When a blood vessel is damaged and collagen in the wall is exposed, circulating platelets begin to slow down, converge, and roll along the vessel wall near the injury. They attach the exposed collagen, which activates them. Activated platelets undergo a physical shape change and become capable of attracting other platelets and adhering to more exposed collagen. This process forms a platelet plug. Activated platelets also secrete chemical compounds from their granules. Alpha granules release von Willebrand factor and the platelet-derived growth factors. VWF aids further platelet adherence and activation, while PDGF helps with long-term tissue healing. Dense granules release adenosine diphosphate, or ADP, and thromboxane, which binds to receptors and other circulating platelets, promoting further activation and creating a chemical cascade. And when we talk about blood clot formation, simultaneously with platelets plug formation, coagulation occurs. This process involves a series of chemical cascades initiated by activated proteins called coagulation factors and clotting factors, which circulate in the plasma in an active state. These factors, often represented by Roman numerals, are activated upon encountering vessel damage. There's two main pathways, the intrinsic and extrinsic, which both lead to a common pathway. For the intrinsic pathway, it is activated by blood vessels are damaged and a collagen is exposed to the circulating bloodstream. Contact with collagen activates factor 12, which activates factor 11, sequentially initiating for factor 9. Factor 9 recruits factor 8 and calcium to form a complex that activates factor 10, thereby starting the common pathway. Factor 13 not 13, sorry. Factor 8 is important in the intrinsic pathway. Factor 11 is an intermediary responsible for activating factor 9, and factor 12 is activated by contact with things in the collagen. When we talk about the extrinsic pathway, it is activated by chemical signals released by damaged tissues outside of the bloodstream. Damage to these tissues exposed tissue thromoblastin. Circulating factor 7 associates with factor 3, and phospholipids exposed by the damaged tissue to form an enzymatic complex. This complex activates factor 10, initiating the common pathway. The common pathway is initiated when factor 10 is activated. Factor 10 then combines with factor 5 and platelet surface phospholipids to form prothrombin activating complex. This complex converts prothrombin into thrombin. Thrombin is a critical enzyme that converts the plasma protein fibrinogen to fibrin. Fibrin forms a dense fibrous network which causes blood to congeal into a gel-like clot capable of plugging damaged areas of the vessels. Within several days, a clot is dissolved through a process called fibrinolysis. This involves an enzyme called plasmin, which completely degrades fibrin. After that, it is activated by an inactive plasma protein called plasminogen, synthesized and released by the liver. Plasminogen is converted into plasmin by various molecules, including tissue plasminogen activator, which is produced by endothelial cells in the presence of fibrin. TPA significantly accelerates this conversion of plasminogen to plasmin. This process initiates the breakdown of the clot. Interestingly, fibrinin activates TPA, thus contributing to its own degradation. TPA can be administered clinically to patients experiencing heart attacks or strokes caused by embolus.